Okay, so we've been talking about the Black Death recently as the precursor, the trigger to the Great Rebellion. And I'd like to go in a little bit into an article that I found about the plague bacteria. And I know this is maybe seems a little off track because we're talking about the Great Rebellion, like the spiritual rebellion. But I do want to talk about how this plague bacteria is able to hide for quite a long time. I mean, decades, and then suddenly recur and infect again. And it's not just in the Black Plague of Europe back in the 14th century that this happened and hiding for a couple hundred years or 600 years, but it has done this in other places around the world as well. And so this bacteria don't usually live so long outside of the host. And they have to find an environment that's conducive for their, for them to live. So the question is, how do they survive then? This article is going to address this issue because they've been investigating that recently. And when I say recently, this article is 2018. So I'm going to read this article. I'm going to interpret it as we go. I'm going to focus in on some things. So if you don't know much about biology or medical issues, that's okay. Just hang in there with me. I will interpret as we go. This is also a way for you to expand your learning a little bit as well. So you may pick up some additional education as we go through these things and I interpret as we go. All right. So as I said, here we go. Plague bacteria may be hiding in common soil or water microbes waiting to emerge. Not in common soil, but common soil microbes or water microbes. So they're actually hiding in the microbes, the bacteria, because bacteria are smaller than the microbes. So let's go ahead and have a look at what this is talking about. Plague is a highly contagious disease that has killed millions of people over the past 1,400 years. Outbreaks still sporadically occur in as many as 36 countries worldwide, which includes the western United States, which is where I'm from. Perhaps one of the greatest remaining mysteries surrounding plague is how and where it survives between outbreaks. And that's one thing I asked about, I mentioned in passing in the last video. Like many other pathogens are the things that cause the diseases. The bacteria that causes plague, Yersinia pestis, remember that name? We talked about that before. It cannot survive for long periods of time in the environment without protection. So it's saying this is one of those bacteria that cannot survive for a long time unless it has some protection. Despite this, plague outbreaks continually recur in many locations. It's not that they occur in one location, then occur in another and another. It's that they reoccur in the same locations. Okay? This suggests that the bacteria are able to find refuge and survive for several years after an outbreak before reappearing seemingly out of nowhere and starting another infection cycle. Understanding where they hide and how they survive and reappear is extremely important for preventing future outbreaks. Okay. Our 2018 study conducted at Colorado State University's Infectious Disease Research Center shows that amoeba, common soil and waterborne microorganisms that eat bacteria, could play a role in protecting this dangerous pathogen between outbreaks. Now, I'm not surprised it's a university in Colorado, because I remember as a kid and a teenager hearing about some outbreak of the Black Plague in Colorado from squirrels. So I'm not surprised at it because I remember from my childhood. But this is a little surprising because the amoeba is a single-celled organism. And yes, it eats bacteria. So how is the bacteria using the amoeba in order to survive? Okay. This relationship may give plague bacteria a place to replicate, to reproduce, and bide their time before conditions are right for another outbreak to occur. Let's get rid of that. Plague has caused, an ancient and mysterious color. Plague has caused 3 deadly worldwide pandemics. The plague of Justinian, we talked about that, killed millions of people in the Byzantine Empire between the years 541 and 750. That's 200 years. And it was actually, it was fairly dormant for 200 years. Next, the notorious Black Death ravaged much of Asia and Europe from 1330 to 1480, killing approximately 30% of all Europeans. And we saw it was 30 to 60%. Most recently, plague reappeared in China in 1855 and spread to ports worldwide over the following century, killing some 12 million people. Thousands of small outbreaks have occurred between and following these events. Part of what makes understanding plague so difficult is its ability to infect over 250 mammals and many species of insects via multiple routes of transmission. Those are vectors. So this plague can infect so many different species of mammals and insects that it is astounding. For example, it can be transmitted through a bite from an infected flea or by inhaling bacteria coughed up by an infected animal. 250 mammals can carry this. I'm sure dogs are part of it. And understand, throughout the Western world, we have dogs in our home. And cats. And cats are even a better vector because cats roam. Cats are outside more than dogs and freely roam and then come back. Not only that, they interact with rodents and with birds and then come back. So they're a perfect vector for the plague to enter the home. So if you're thinking about getting rid of an animal, I would start with the cat. And I would get rid of the cat in your home. Just ban cats from your home. Of course, then you might have problems with rodents. And then you've got fleas on the rodents. So either way, you can end up having a vector in your home. If you've got a dog and you want to keep the rats and mice away, a good one is a dachshund. A dachshund is, it can be as fast as a badger. We had one and it was a great mouser. It would catch it. Boom. Just catch it. Better than a cat. So yeah, you could use a dog, and dogs are more trainable and you can contain them more in the home. So I would say, if you're going to get one, get a dachshund. That's a good one to keep in the home. But not a cat. Plague outbreaks also occur across very diverse environments. They range from prairie ecosystems in the western United States. You can see the big red, like 3/5 of the United States is red. That's all the prairie lands, including the Rocky Mountains. So it's not just prairie lands, but it goes all the way across. That's the area I grew up in. Exclude that very, very middle part, where Texas is and up to North Dakota. Everything west of there is where I grew up, mostly. A little bit in the east. They range from prairie ecosystems in the western United States to highland forests in central Madagascar and temperate deserts in western China. The fact that few characteristics unify all of these regions may indicate that plague bacteria use different survival mechanisms in each location. However, one unifying factor is the presence of amoeba in the soil. And we know amoeba eat bacteria. They consume them. Now, the question is, can they digest this bacteria? So you see, all these red areas that's the primary locations of the plague today, as of March 6, 2016. So you can see. And not so much of it in the southern hemisphere. It's kind of scattered into small pockets. But in the northern hemisphere, it's the mid and western United States. That is the second biggest area. The other biggest area is Central Asia, spreading across Central Asia. Now, see, it doesn't quite get into Ukraine here. Here's the edge of Ukraine right there. This is Ukraine, this country right here where I'm spreading my cursor across. And just outside of Ukraine, that's Russia. Because Russia comes down here all the way to the Black Sea. Like Sochi is down here. And so you can see that it stretches all the way across Central Asia, the steppes of Central Asia, into Western China, where the Uyghurs are. That's a Muslim people group. And then you got it down here in parts of India here, about a fourth of India. Abundant hosts. Amoeba are single-celled microorganisms that live in almost all soils and water bodies across the globe. This is what they look like. They don't really have a specific shape, but they do have some very consistent things where it has like legs sticking out here and there, and things that look like a gut or whatever. But it's kind of an amorphous shape. There's no particular shape. Whereas bacteria have this, some bacteria have this, capsule, this pill shape. They feed on bacteria, but scientists have discovered that some bacteria are resistant to being digested by amoeba. There it is. Some bacteria refuse to be digested by the amoeba, even though it's ingested. It's ingested, it's taken in to the digestive system, if you want to call it that. It's just a single-celled animal. But it can't be digested. Interestingly, they include plague's most closely related ancestors, Yersinia pseudotuberculosis and Yersinia enterocolitica, I think, living in the soil alongside amoeba, required these bacteria to evolve ways of being, to avoid being eaten. Okay, that's their explanation, is evolution, right? Given this evolutionary history, our research team hypothesized that when plague evolved from Yersinia pseudotuberculosis approximately 10,000 to 40,000 years ago, it may have retained the ability to survive inside amoeba. Well, don't just guess. Just do an experiment and see. Natural disease incubators. An incubator is like where an egg is being prepared to hatch into a chicken. The chicken's growing inside the egg. So to test our hypothesis, we collected soil samples from prairie dog burrows in northeastern Colorado because prairie dogs are known hosts of plague. So they're so cute, but they carry the plague. This involved finding prairie dog colonies that were experiencing a plague outbreak and inserting a long, flexible probe into burrows to collect samples from deep within, while avoiding potentially infectious fleas emerging from the burrow. Yeah, it's dangerous. After isolating amoeba from the soil in our lab, we identified 5 species to use in future experiments. Okay? 5 species. So, of amoeba, right? The next step was to determine how plague bacteria interacted with the various amoeba species we identified. In a special high containment laboratory designed to prevent dangerous pathogens from accidentally escaping, we combined amoeba and various strains of plague bacteria obtained from the U.S. Centers for Disease Control's laboratory in Fort Collins, Colorado. We used a genetically altered strain of plague that fluoresces neon green to determine if and when amoeba were ingesting plague bacteria by viewing them under a high-powered microscope. And here is a picture. Okay, it says fluorescent plague bacteria inside amoeba. Okay, scale bar indicates 30 microns. So this is 30 microns. I don't know if you've used a microscope before, but next we used a transmission electron microscope to obtain even more detailed images of the inside of infected amoeba. They're not infected amoeba. the amoeba swallowed them on purpose, trying to eat them. This revealed that the plague bacteria were alive and possibly replicating. To confirm this, we selectively cracked open the infected amoeba at different time points to compare the number of bacteria inside. Our results are the first to demonstrate that plague bacteria are able to survive and replicate inside amoeba. YP is Yersinia. It's the bacteria, right? This is the plague bacteria here. You got it inside these like vacuoles or these kind of vacuum pockets. You also have them just inside amongst everything else. The next challenge is understanding how long plague bacteria can survive an amoeba. A part of an amoeba's life cycle includes transforming into a cyst, a form in which it can lie dormant for up to 20 years before it reanimates and resumes eating and multiplying. So the amoeba is able to transform itself into this kind of hardened, dry, dormant state that's able to last for up to 20 years. And then after that, it reanimates itself, absorbing moisture and coming back to life, and then starts eating and multiplying again. So the question is how long, right? How long can the plague bacteria survive an amoeba, right? Considering the cyst stage as well. This enables it, the amoeba, to survive during adverse environmental conditions such as extreme temperatures or drought. If plague bacteria can survive inside dormant amoeba cysts for many years, this could explain how and where they persist, misspelling, should not have the S on the end, between outbreaks. So it's interesting, isn't it? So they may have found the location of the plague when it's not infecting humans and other animals and insects. It's lying dormant, so to speak, inside of amoeba that are in the soil and the water. So the black plague's in the soil or the water, but inside the amoeba in the soil or the water. Amoeba as disease training grounds. This is really interesting. Amoeba are already recognized for their potential role in protecting, amplifying, or guiding the evolution of over 225 other bacteria, viruses, and fungi. Famously, it is hypothesized that they played a role in the first known outbreak of Legionnaire's disease in 1976 by providing a protected space for Legionella bacteria to multiply. Amoeba can also act as transport vessels for pathogens, enabling the bacteria to enter and infect new hosts. Some scientists hypothesize that amoeba can guide harmless bacteria to evolve into dangerous pathogens. The reasoning behind this is that amoeba are very similar to macrophages, the white blood cells in mammals that are responsible for finding and killing invading bacteria. This is what's so astounding. They say that the amoeba and the white blood cells that are macrophages are very similar. And those white blood cells, it says, are responsible for finding and killing invading bacteria. If harmless soil bacteria evolve the ability to survive and multiply within amoeba, then they might also be able to do so in the white blood cells that make up our immune system, thereby becoming new human pathogens. So if these bacteria can develop some kind of system for surviving and multiplying inside of the amoeba, then it's quite possible that it can use the same skills to do the same inside of our white blood cells once it enters into the human body and the white blood cells try to consume it. So they might have a way of preventing it from consuming it because they're very similar to the amoeba. And if they developed a way there, maybe it's transferable there as well. The idea of amoeba acting as training grounds for the evolution of dangerous pathogens like plague conforms with what scientists already know about human plague infections. In human cases, plague is engulfed by white blood cells Okay, so the plague bacteria is engulfed by white blood cells, but has evolved a way to avoid destruction by escaping the portion of the cell responsible for digestion. So the part of the cell that actually breaks down the bacteria, it avoids that part. Okay, then it multiplies inside the white blood cell, avoiding that part that is responsible for breaking the bacteria apart. It multiplies inside the white blood cell before exiting and disseminating throughout the human body. So it uses the rest of the white blood cell for a location to multiply. And then after it exits that, then it infects the rest of the human body. This process is nearly identical to the process our research team observed in amoeba. Did this ability to avoid destruction and multiply within white blood cells, which is found in many human pathogens, arise from ancient soil-borne bacteria learning to exploit amoeba? Perhaps practice really does make perfect. Pathogen harboring amoeba could be serious public health threats, since we currently have no way to efficiently monitor them in the environment or predict when they might release infectious agents. They may also pose a biosecurity threat that a hostile power could use to disperse existing pathogens or create new ones. These people are so paranoid. They are so paranoid. This is the illness that marks the American people right now, is paranoia, more than any other illness. Paranoia. And among Christians too, you American Christians who are watching this, stop being paranoid. Trust the Lord. It's okay to study and learn things, but look at the leap that they made into science fiction. Could pose a biosecurity threat. How? They made ambiguous statements after that are unrealistic. You got amoeba in the water and the soil. How are you going to spread amoeba in the soil? Through the water, maybe. But you can spread anything through the water. Why amoeba? So the bacteria will lay dormant for 20 years? Why? Why not just disperse the bacteria and infect the people? They're not thinking. They're just trying to take whatever they're talking about right now and then blow it out of proportion and use it in their paranoia and their conspiracy theories. And then this paranoia and the conspiracy theories are then fuel to create a dictatorship and a police state. So don't participate in that. If you're a Christian, do not participate in that. Stop being paranoid. If you have the persuasion, the faith in Jesus Christ, you don't need to be paranoid. Paranoid of what? If something's going to happen, it's going to happen. And so what? Well, you need to be prepared in case it happens. Well, if you get this knowledge and you have the option of being prepared, be prepared. But don't walk around being paranoid about everything or looking for opportunities to create a paranoia in your head. There's no place in the Christian life for paranoia. Now, that having been said, this answers the question of where did the Black Plague hide? During those times in between the outbreaks in Europe that we were talking about with the Black Death, where was it hiding? Like I had said before, I suspect that the Black Plague outbreak in the 14th century came from the bacteria of the Justinian outbreaks in the 6th and the 8th centuries, 6th, 8th, 14th centuries. And it was dormant. Well, it may not have been dormant exactly. It could have been passed through insects and mammals, 250 mammals, can transmit it. Insects can transmit it, so it could be living in these organisms, and in between in the soil as well with the amoeba. So that explains it right there. That answers our question. Remember to subscribe down below and like the video and share it on your Facebook and other social media. And then make a comment, whether a question or a comment. We read all of them, and we try to respond to all. Get on over to our website, The Rooted Word, and start reading the translation and also the articles we've posted. It's at therootedword.com, therootedword.com. And may the Lord bless you as you seek Him with all your heart.